Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. In present-day neuroscience, a commonly used research approach is to record activity from hundreds or even thousands of neurons, either with electrophysiology or with optophysiology, and then use various statistical methods such as principal components analysis, PCA, or other so-called dimensionality reduction techniques to attempt to gain insights into the actions of the underlying neural networks. In 2017, the paper Could a Neuroscientist Understand a Microprocessor made some stir in our neuroscience field, since it raised serious questions about how much we can expect to understand of the brain from this research strategy, at least when applied to mammalian brains with millions or billions of neurons. In the paper, the authors Eric Jonas and Konrad Kording applied the same neuroscience approach to a computer microprocessor where a transistor can be argued to be analogous to a neuron. Unlike the brain, the microprocessor is fully understood. After all, it has been made by humans. In their study, they found that some recordings from transistors in the microprocessor could qualitatively resemble what is seen in neuronal recordings. However, their conclusion was that you nevertheless couldn't understand much about how the microprocessor worked from these recordings. So the uncomfortable question raised by this study was, how much can the present way of doing network neuroscience, that is statistical analysis of large-scale neural recordings, be expected to teach us about how the brain works? One of the authors of the paper, Konrad Kording, is the guest in today's podcast. Partially motivated by the results from his microprocessor study, he now argues that we should attempt an alternative approach and focus on the tiny roundworm C.elegans with only 302 neurons. In a perspective paper published on Archive in the fall of 2023, he and 2030 co-authors suggest that we should attempt to reverse engineer the neural system of this tiny organism. Here they argue that modern optophysiology techniques can be used to stimulate and record activity of each of the 302 neurons. Further, this technique should be able to scale to hundreds of thousands of experiments across a large population of animals. Modern machine learning-based model training should then next be used to make a mathematical model that allows for accurate simulation of the behavior of C. elegans for all kinds of stimulus conditions and brain states. In the podcast, we first talk about the 2017 microprocessor paper before moving on to the motivation behind and specific plans for reverse engineering of C. elegans. As for all episodes, information about the episode and relevant links can be found at the webpage theoreticalneuroscience.no. If you want to support the podcast, go to patreon.com/theoreticalneurosciencepodcast and choose a membership of your liking, for example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast, where you can watch the videos as well. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Okay, here is Konrad. So, welcome to the podcast, Konrad Kording.
Great to see you, Gaute.
Yeah, so very good. I think we met when we were both very small. At least I was very young in neuroscience. I think it was in Lithuania in 2000 or something.
Yeah, I was a young PhD student. It was awesome.
But I still remember you as sort of like quite eager and active, so that you had you had a lot of energy then, and you seem to have a lot of energy still.
You already had a lot of energy back
Yeah,
then as well. Yeah,
that's maybe true. No, but that's good. So at least we both made it, in the sense that we are able to keep on working with science. So I guess we are fortunate in that sense.
Indeed. Yes.
Yeah, so good. So I mean, today's podcast is actually about your very interesting plans, I think, for this reverse engineering of the nervous system of this very small roundworm, C. elegans, only 302 neurons. But first, I would like to talk about this. I remember you had this paper that came out in 2017, and you said something like, if you ever go to read one paper by me, read this one. Because it was about sort of, could a neuroscientist understand a microprocessor?
Yeah.
So what was that about?
Well, it's so when we do neuroscience, there's a big problem, which is it's usually difficult to know if you're right. Now, if you're an engineer building bridges, it's very easy to find out that you've been wrong. Like the
Mm.
bridge falls, caves in, and everyone dies,
Mm.
and you're fired as a
Exactly.
bridge builder.
Exactly. Yeah.
The big problem in neuroscience is no one gets fired for any reasons because no one knows what's right and what's not. Now, like
Mm.
if you do neuroscience, you usually
Mm.
do some recordings from brains, but the recordings themselves aren't interesting to us. No? We
Mm.
want to understand how the brain works. So we
Mm.
get some recording and then we say something about brains. And no one knows if what we say is actually correctly deriving from the measurements that we have.
Mm.
And
Yep.
that is like the starting point for me. And so then came that paper with Eric Jonas, who's like this absolutely brilliant guy.
Mm.
And what that paper said is, let's take a system that we actually do know. In that case, a microprocessor, the 6502. It's a classical microprocessor.
Mm.
Probably you used it when you first started programming.
Probably yes. Yeah.
and and so then let's take this as a device that we understand and see if we do neuroscience with that device.
Mm.
Is what we learn about it really right?
Mm. Yeah.
So the idea is we want to be no, it's technically part of of a branch of philosophy, epistemology. Like,
Mm.
what is it that we can know about the world based on the
Mm.
measurements that we take?
Yeah. And
And
when
the
you say just one, when you say neuroscience, you mean maybe like the most common way of doing neuroscience of now recording data, maybe now with these techniques with many neurons, many spikes from many neurons, and doing some kind of data analysis on it. That is sort of what you mean, right?
Yes, let's go through the logic as we often use it in
Mm.
neuroscience. You know, like
Mm.
what people do. In fact, what I was involved in during my PhD, I wasn't very good at it. That's why you don't read many papers about it. But back then, the idea was we're going to stick an electrode into a brain, in some area, maybe primary visual cortex, and then we're going to show some stimuli in the outside world, and then we see what happens to the neurons that we record from, and maybe we find that the neuron is most active. If we show
Mm.
stimuli of some direction, and then we're like, oh, therefore, the role of the neuron in the computation in the brain is to tell us about the presence of that stimulus. No? And in
Mm.
primary visual cortex, we all know it, like oriented bars and gratings were the big thing for a long period of time. So basically, primary visual cortex must be a machine that tells us about bars and gratings.
Mm. Yeah.
And then the question is that logic right?
Mm.
And we are trying to poke holes into that logic in the case of the microprocessor.
Mm. In some sense, you're looking for a benchmarking system to test your where you know the ground truth.
That's right. Yes.
Yeah. Mm.
No, and if you look at the field like machine learning, it got really, really good once we had good benchmarks.
Mm.
So,
Yeah.
and you can say probably the
Yeah.
same thing is true for branches of science. Basically,
Mm.
if we know what's good and what's not, it's easy to do good. But
Mm.
if we don't know what's good and what's not, it's kind of like very difficult to
To
know
make to
what's good.
make to make progress, right? Because you don't really know if you're doing better or worse, right? Yeah.
That's
Absolutely.
right. Yeah.
Mm. Cool. So what was the and then I read this paper just before recording this. You seem to know a lot about sort of like, are you electrical engineer or maybe you or Peter or that no Jonas, right? What was his
So
name?
Yeah, so Eric Jonas
Eric Jonas, sorry.
is an impressive engineer on top
Mm,
of being a great neuroscientist. And
mm,
so Eric had just a lot of background on electrical engineering.
mm. Yeah, so he could sort of, and then okay, so then, yeah, that was like a side remark. I was just impressed by all these registers and buses and all these things that the electronics students learn.
But
So
I've also always been fascinated about it.
yeah,
Like, it's amazing that our tech world is built on microprocessors and
mm,
memories and all that.
yeah, yeah,
And kind of getting a feeling for how they work is just beautiful. And so
yeah.
I
I
always
agree.
enjoyed reading on electronics.
Absolutely, no, that's true. It's a very, very impressive technology, and we almost take it for granted, right? Because it's sort of it's
We we,
everywhere, and yeah,
we do. It's in
yeah,
my phone, and my
yeah,
phone is would have been the world's best computer a
yeah.
relatively short period of time
Yeah.
ago.
Yeah. Yeah. No, it's sort of quite. I actually read this book called The Maniac about John von Neumann, who came up with many. Well, he was really like a multi talent, or so he also came, of course, with this. I guess with the most common architecture for doing this, which still is used as in normal computers. I think in most computers.
Yeah, I mean, like, we are building very parallel systems, of course,
Yeah,
but
that's
like
true.
a lot of the
Yeah.
like the von Neumann ideas are still all over the
Mm.
theory
Mm.
of
Yeah.
machine learning and the theory of computing.
Mm. So what was okay? So what was I mean, your finding that you tried to use this typical data analysis techniques that is commonly used by neuroscientists, like whatever, PCA or correlations and all kinds of things, right? And on the some sense the data from this microprocessor.
Yeah, like, I think the upshot is we produced lots of really wonderful looking
Mhm.
figures that would have been very publishable in neuroscience.
Yeah,
But these figures didn't contain the deeper meaning that we'd be looking for.
yeah.
And maybe to step you through it, now what we did is we asked, um, how does the activity of a transistor in the microprocessor relate, say, to the brightness on screen? So it turns out that
Mm,
these old microprocessors, they basically write pixel by pixel. So
mm.
it makes sense to kind of say, well, maybe there's like transistors in the microprocessors that are related to the brightness of the
Yeah.
newest thing.
And the transistor is a little bit like the analogous to the neuron.
That's right. Yeah, no, like the transistor, just like a neuron, is a time varying device. It's kind of mostly binary. I mean, it switches up and down, while neurons are mostly like they're silent, and then comes the spike. But
Mhm.
at some level, they felt like they were like comparable devices.
Mhm.
And
Mhm.
then we can apply like what neuroscientists do. And we basically then make curves where you on the x-axis you have
Mhm.
the brightness on the screen. On the y-axis you have how often
Yeah,
how often that
that's
transistor switches.
like
And indeed we found like very strong tuning for brightness.
like tuning curves.
Tuning
Sure.
curves. Neuroscientists would be very happy. It's only
Yeah.
that the neuroscientists would immediately go and jump to that conclusion. Oh, because brightness affects that transistor, the
Mm.
transistor must be a brightness
Yeah,
signaling
exactly.
device. And it
Mhm.
turns out that, of course, the great thing about the microprocessor is we know what the transistors do,
Mm.
and they don't transition brightness. They don't transmit brightness, but it might be that it's a transistor that is used as part of adding numbers. Maybe
Mm.
it's like the sixth bit
Yeah.
of register number three that does something, or maybe it's a part of the circuitry that transforms basically commands into
Mm.
electrical signals, the so-called decoders. Um, so what we found is that it's really not that the insights that we'd be getting in neuroscience would be correct when it comes to the microprocessor.
Mm. Yeah. So exactly. So then that was sort of like a bummer message, right? That it's sort of not really the way to go. So what was the response to this in the community,
So
neuroscience community?
the response was remarkably mixed.
Yeah.
So parts of it, there were a lot of people that were unhappy with the title. So the title
Yeah.
was "Could a Neuroscientist Understand a Microprocessor?"
Okay.
which was a quotation of an earlier paper, which is "Could a biologist understand a radio?"
I
Where
see.
they basically, in biology, there's a common trend of trying to uh, understand systems by destroying pieces. You know, like we all know, like you have like knockout mice, and like you take out a gene, you're like, okay, does it still walk? Yes, it
Mm.
does. Does it still like talk? Well, no, it never talks. But like, kind of, it goes through these things,
Yeah.
and that's the logic of biology. And there was a wonderful paper where basically Zalapnik was asking about like, could we really understand a radio if we would treat it in the same way as we treat biology? And he pointed out that not.
Mm.
Now, as opposed to him, Eric Jonas went all in on that. He basically said, okay, now let's actually do that. So Lapniks is more a Gedankenexperiment. It's
Mm.
more like, what would happen? Would we understand it? No, we wouldn't. Whereas we went a step further. But like the fact that we phrased it like that, could a neuroscientist understand a microprocessor? A lot of
Mm.
people viewed this as us dissing neuroscience. But
Okay.
we love neuroscience, no? Like
Yeah,
we're
of course.
neuroscientists
Yeah.
ourselves.
Yeah.
So I think that there was a bit of a misunderstanding there. It's
Mm.
just we were giving credit to people who came before us who did. I mean, I'm just such a fan of this. Could a biologist understand a radio kind
Mm.
of a message? It was beautiful and
Yeah.
very inspiring to me.
Yeah.
So there was some pushback there. There was pushback where biologists said, "But we use knowledge about biology. Your microprocessor paper like doesn't use knowledge of microprocessors." But
Mhm.
then, if we are honest as neuroscientists, we aren't using knowledge, deep knowledge of biology most of the time.
Mm.
Um,
And particularly in statistical analysis, I guess. I mean, that if there's no like biophysical constraints on the data analysis, then I guess you don't really use it, right? I mean, at least.
Yeah, I mean, like just like a few weeks ago, we had a little paper in PNAS about people using dimensionality reduction.
Mhm.
And neuroscientists believe that if they run things like PCA on data, big domain in the MRI literature, big domain also in the spiking literature, and they use more sophisticated version, JPCA.
Mhm.
There's a whole host of approaches that at some level it reveals how the systems work. And I
Mhm.
think the microprocessor was a first example of what the problem is. But the thing is just if we run PCA, we're going to find the dimensions that are carrying a lot of variance. We aren't going to that is not a dimension that tells us how a system works. Now,
Mhm.
if we apply it to the microprocessor PCA, we do find something that's real. No? Like there's a clock in there, and that clock carries a lot of variance, and you can describe a lot of variance in terms of the clock. So the
Mhm.
first principal component or second or something like that carries a lot of the clock signal. But that's not the only thing that carries clock signals, and it doesn't only carry the clock signal. And this thing that the things that you find with algorithms doesn't reveal how biology works, but reveals things that are correlated with a lot of variance
Mhm.
is a universal problem in the field.
Yeah. So then with like this question of sort of revealing how biology works, what kind of requirements would you have for an understanding, right? That it's what does it mean to understand how something works? That's a separate podcast about tell
Yeah,
people, right?
yeah, and that is extremely difficult. Neuroscientists usually say what they are after is a mechanistic understanding of
Mhm.
brains.
Yeah.
And if you dig a little,
So
in
what
fact,
you mean then is like so what do you mean by mechanistic understanding? Just to make sure that
so
everybody
so
is on board on this
yeah,
important
so
point.
the way most neuroscientists, so they usually don't talk about what exactly they mean with it, which is normal. In
Mm.
most clubs, don't define exactly what it takes to become a member of it. But suddenly, if you're not into a mechanism,
Yeah.
then neuroscientists will be very unenthusiastic to fund you. And their definition is usually, and I'm saying that usually because no one really defines it. There was a very nice paper um, uh, by Danny Bassett and Lauren, and blanking on the name right now.
Mm,
Just just came out in Nature Reviews Neuroscience. It's a wonderful paper, and they focused on mechanism. But like, if you zoom out a little, what they mean with mechanism is a understanding of a causal effect,
mm,
along with understanding the chain that implements the causal effect. For
mm,
example, causal effect, if I have two neurons that have a synapse in between, I make the first one spike, it produces an excitatory influence on the postsynaptic neuron.
mm,
The underlying biophysical mechanism is there's a synapse in between that dumps
mm.
glutamate into the
Yeah.
extracellular space with receptors on the other side that open ion channels. Now,
Mm,
like, that's like the quintessential mechanism that they're looking for. The important thing is there's the two components. The one of them is I want to be able to causally know what does one element in my system do to the next one.
mm,
And the second one is I want to understand how it does so.
mm. Yeah.
Uh, and that is that I think is what neuroscientists would say they're after if you quiz them very hard on
Yeah.
it.
That's at least a reasonable goal.
So, but let's think through that reasonableness of the goal. So,
Mm,
if you're a slice physiologist, no, you're like you take a small part of a mouse brain, you cut it very thinly. There's just like a handful of neurons alive in that slice, and you patch clamp the two neurons. Then it's very easy for you to get at that causality because you make the first neuron spike and you record from the other one, and you see what happens there. So for slice physiologists, this notion of mechanism is very obtainable. But if we are fair, most of the neuroscientists want to understand humans, or at least mice, as as a proxy for humans.
yeah.
And if what you want to understand there is like that, then you're in trouble, no? Because you will record, there's Stevenson's law. Every 6 years or so, like the number of simultaneously recorded neurons doubles. But
Mm,
hello, we're like at about 10,000, and there's still, which is still, well, a tiny piece of, say, the 100 million that we have in mice,
mm,
or the 80 billion that we have in humans. So we're looking at this tiny piece of a system, but then we want to tell these causal stories about how pieces in the brain influence one another. And that is something that that people often do, this idea of functional connectivity. Like, I take the 1,000 neurons that I record from, and I fit to the data an interaction model that kind of pretends that the world only consists of 10,000 neurons, or alternatively 10,000 dimensions on which I do PCA, if I talk about a lot of things happening now. So
mm,
we have this tiny piece, but what we want mechanism wise is really how they interact with one another. And of course, that doesn't work.
mm,
And because it doesn't work, modern neuroscience has all these like weird ways of bridging that gap. Like, we have mechanism, and we convinced ourselves that anything that isn't in terms of mechanism shouldn't even be published. It's not neuroscience.
mm,
And then we have these techniques where we stick a couple of electrodes into a brain that has a lot of neurons. And so the question is, how do we bring what we can have, which is record from a couple neurons, with what we want to have, tell us how like interactions happen in the system. And
mm,
so what do? We basically either do what's hypothesis testing. Let's imagine there's only three ways how brains could work, and choose which of the three of them is the best. Great, works no problem. It's just that like the space of meaningful models is just so much larger.
yeah.
And the other one is, let's just ignore that we are only recording a tiny bit, and use, if you look at the functional connectivity or effective connectivity, there's an awful lot of research that just basically pretends that correlation is causation. And then
Mm.
for them, life is very easy. If
Yeah.
correlation is causation,
Yeah.
measuring correlation between the thousands of variables that we record from is very easy, whereas causality is very, very
Yeah,
difficult.
because then you have to stimulate in a controlled way or something, right? We're coming to then you have
Yes.
to sort of control the stimulation at the neuron level.
So kind of let's do a step back. Like, why
Mm.
is it so difficult to know how causality works in complex systems? Um, Correlations are easy to measure. The problem is that there's confounding. If I have two variables and they could be correlated because they interact with one another directly, but it could also be that there's just a third variable that affects the two of them. And
Mm.
then there's, of course, clever people who say, well, if there's a third variable that affects them, why don't we correct for that? And indeed,
Mm.
there's a branch of data analysis focusing on that. But then there's the problem of what we call collider, which is if there's a variable that doesn't affect the two of them, but is affected by the two of them. And
Mm.
if I correct for a confounder, the problem gets better. If I correct for a collider, the problem gets worse. And
Okay.
in reality, in the brain, almost everything is simultaneously confounder and collider, and therefore it's entirely unclear if there's probably no meaningful statistical ways of
Mm.
dealing with it.
Yeah. Mm. So that's not the thing. So then I guess I mean after the I guess I mean now is it right to say that I mean your the insights or the conclusions from this paper motivate, because I guess one of the reasons why people were sort of not so happy to hear about it was that there wasn't any clear alternatives, right? How what can we do then, right? I mean, should we all go home and and sort of so maybe that was the reason. So
Yeah,
so
yeah. Look, like what I've been arguing in that paper and a lot of follow-up papers is
mm,
neuroscientists want to get at causation. They want to get at how it works,
mm,
but the methods they have fundamentally doesn't allow revealing how it works. And now with this, neuroscience is really in a problematic situation, and people don't like hearing that the field has a fundamental issue. In particular, they don't like hearing it because as a field we get addicted to that causal mechanistic language.
mm,
It's so deeply in the field that you can't publish, you can't get grant funding. It has all those problems.
yeah,
And so if you then say, well, hold on, you're not delivering on what you say you're delivering on, people really don't like hearing it.
no, no,
And with the big difference in ages of how people responded, a lot of senior people who basically said, like, how dare you?
mm.
In fact, like, this will reflect horribly on your career, Konrad. Are you sure you know what you're doing? And other people, a lot of young people that, like,
yeah,
you see, like, a good person knows all that, feels all that. And, like, we
yeah,
can't suppress it. A
yeah,
lot of young people who were basically like, look, this is great that you're doing
mm,
that because I always felt like that.
yeah,
But
mm,
people were kind of telling me that I'm wrong about it.
mm, mm,
And so the responses are very bimodal on that, with
yeah,
people absolutely loving the paper and people
mm,
absolutely hating it. And a
mm.
lot of people taking a somewhat reflected position where they're like, okay, I want my students to think about it, but Connor's still a horrible person.
I see. Oh well, that's well. This is I don't know. Obama said this is what change looks like. maybe, maybe. Don't know. But now,
Hopefully.
yeah. But now at least sort of you have, I guess you're like the what we yeah this idea of reverse engineering, like one small system that which are sort of like one of the people spearheading. I think there's like at least something came out on bioarchive or something in the fall of last year, 2023. So that was maybe like for you like a way to try to go beyond or to deal with this problem.
Yes. So the problem with causality, there's two problems with causality. The first one is you need to stimulate. The other problem of causality is a power problem. If I want to see what's the causal influence of, say, one neuron on
Mm,
another, and there's lots of neurons, then everyone will have a very weak influence. And it's going to be very difficult for me to know how strong is each of the
So I mean, like
influence.
statistical power, right? So
Yes,
not, yeah,
statistical power.
yeah. So you don't
And
have enough not enough data compared to the number of unknowns or something.
that's right.
Yeah.
And that's why I'm now so excited about C. elegans.
Mhm.
So C. elegans has 300-ish neurons.
Yeah. Yeah.
The
Tell about
hermaphrodite.
C.elegans. It's been
So
like, yeah, it's been like this big model system since I guess Sidney Brenner is like one of the people who developed this, like the what's called the DNA protein kind of what's called the molecular.
oh, it's been a wonderful model system and almost always ahead of the curve. That
Okay.
includes it had the first known connectome. They basically the connections are very similar across the across worms. So
Mm.
which neuron contacts which neuron is relatively
So
conserved.
exactly.
We know we have a census of all cells. Now we know which cells are there in C. elegans, and it's the
Mm.
first animal I believe that has that property.
Mm.
It it's been the first that was entirely sequenced. So it's been a wonderful system uh, that's been extremely productive
And
in
it's
lots
really
of
this
ways.
little worm, isn't it? What is it really?
Yeah, so let's talk about what it does. So it's a small worm. It's about 1 millimeter long, about 50 microns thick.
Mm.
It hunts for bacteria, and a lot of people don't give it the respect that it deserves because
Really?
C. elegans, to say it in a funny way, does almost everything that you or me does, minus the talking. So let's let's talk about the cool things that it
Yeah.
can do.
Uh,
It if it's hungry, it's looking for food. It avoids predators. It avoids bad temperatures. It avoids noxious chemicals in the
mm.
environment.
Mm.
It finds mates. It does mate and have babies.
Ah.
It lays eggs. It
Because they said it was that's like it changes sex during its life. Is that how it is? No. How is that?
so it just means that it can reproduce without
Oh,
males,
that's what it means.
but it
Okay.
can also reproduce with males. So
Ah,
there
very
exist
flexible.
males, and at least in C. elegans, it looks like being a male requires a few extra neurons.
Mm.
But the hermaphrodite is the model system that is most studied, as opposed to the males.
Mm.
Um, you can argue if the males fight with one another, they do at least do things that look like fighting,
I
or like
see.
that have a similar computational role as fighting.
Mm.
Um, and it does other crazy things, like imagine you're C. elegans, you're a little colony, bunch of worms locally, and the food's gone. You ate all those sweet bacteria.
Mm.
What are you gonna do? Well, it turns out that they can basically they exist in multiple forms, with dower being one of them that can last for a longer period of time. Basically, if you're like run out of food, you basically try and put yourself into a good longevity situation. And if really push comes to shove, they can apparently try and get picked up by insects and being
Oh.
put to a new place. So
Oh, really?
they go to a place, charge themselves, and flip go onto
Oh,
an
really?
insect and fly away.
So
So
they immigrate.
they emigrate,
Yeah,
exactly.
exactly.
So in any case, what I just want to say is they have like this incredibly rich set of behaviors that's like
Mm.
really quite interesting if you come from the mammalian systems, where they
Mm.
really like can do a remarkably they 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 have a remarkable set of possible behaviors.
Mm. And these neurons, like 302 neurons you mentioned, and it's like 900 cells altogether or something. Is it
Something like that, yeah.
something like that? So 302 neurons. Do they fire like action potentials, normal action potentials, or what do they really? What's the electrophysiological properties?
So there's a small number of neurons that, if I remember that right, do spike.
Mm.
Most of them don't spike, but are analogous.
Mm.
I mean, like you have a good a fair bit of analog neurons in your body that
Yeah,
don't
like
spike.
the retina,
Uh,
for example.
Retina has plenty of them,
Yeah.
exactly. And you can say spikes, if you think about it computationally, a really great way of transmitting signals over long distances.
Mhm.
And the worm is just like a little bit smaller, so it doesn't need spikes quite that much.
Local communication. Yeah, I see. Yeah. So okay, so that's
But
they're also quite challenging to record from electrophysiological, isn't that? There's something that they it's difficult to
yeah, so
patch them, aren't they? Or
exactly, you can patch it with people like Miriam Goodman
uh,
doing those things, but the easiest way to get there is by doing calcium recordings.
hmm.
So what we can do is we can have basically calcium dyes, and it's possible to localize them into the nucleus. And the reason why that's attractive is you basically produce more or less point sources of light. And point
Okay.
sources of light make it much easier to solve the inverse problem. Now, like, you will ultimately want to do a transition of the videos that you're going to record if you have
Mhm.
an optical situation into
Yeah.
basically temporal traces of the activity of every neuron at every
I see.
point of
Mm.
time.
Yeah. So now you're jumping a little bit ahead because now we're going into the reverse engineering thing. So, but we're coming there. So what's the? So you're going to. So you have this, well, 302 neurons, and you say something. You said that the connections between them are sort of quite standardized. I mean, how, or I mean, that's probably it's not. I mean, one thing is whether they are connected, and I guess the other thing is the whatever synaptic strength. And I guess that's have they done a lot of EM or what's known
So,
about this?
so we don't know how well conserved the electrical influences are. There is
Mm,
some stimulation work from Andrew Leifer at Princeton who that does suggest that there's a fair bit of conservation, but that there's also some variance across individuals.
hmm.
And I think I know you don't want me to talk about reverse engineering already,
Well, now
uh,
I can start.
but basically, if all worms would be the same, it would be, from a statistics perspective, wonderful because we could just average data across them. Now, you
Mm,
could say there is something slightly weaker where there exists variance across individuals, and the variance across individuals just kind of makes it so that they do different things. Now, like, you can say, to be evolutionarily successful, it's good if you have some C.elegans that take high risk and get high
mm.
reward, and some
Yeah,
of them that are much more conservative and kind of
yeah.
like have that whole distribution. And in one view, you can say that that variance that we have in the connectome and in maybe the strength of the synapses that we have fulfills that role. But it's also possible that there are feedback circuits that make it that it ultimately lies on a lower dimensional manifold. This is
Mm,
something that we don't quite yet
mm.
know, and it
Yeah.
ultimately makes a big difference if you want to
So,
understand how it works.
has it been done a lot of electron microscopy on
Uh,
the C aliens or?
yes, so there exist EM based connectomes
Yeah.
of I believe we have reasonably complete connectomes on like three worms that are published. We've
Mm,
done some analysis which suggests that at least the rough location of synapses is relatively conserved across individuals, not
mm.
perfectly,
No.
but at least it's strongly correlated across them.
Yeah. Mm. Okay, now to reverse engineering. So the goal is to make some kind of a like a mathematical model representing all these 302 neurons and connections, right? And it's sort of like a
Yeah.
digital something. Now we use this term digital twin. That's sort of quite popular these days. So it's like a digital twin to a real aliens or.
Yeah, so maybe let's first talk about the digital twin term. So,
Mm,
in large parts of neuroscience, when they use the wording digital twin, they mean something that has the same correlational structure. So, what we
mm,
want is not a digital twin in that sense. We want a digital twin that is causally identical. So what I want is, let's say we have the 302 neurons, and what do I know, like 10,000 synapses between them. I want to know for each of the synapses what are their electrical properties. Now, what's their timing? What's the strength?
Mm,
What are their maybe nonlinear properties? And if I had all those causal components of the
mm,
system, I could put them all together into a simulation of C. elegans. And if I identified correctly what the neurons do, then that simulated C. elegans should behave in the same way as the real C.
mm.
elegans.
If
And
you say stimulated with some kind of food molecule, something like the normal stimulation that they are exposed to.
yeah, so there's so if you want success for these simulations, for me has three components.
Mm,
The first one is a correlational, where you could say, I want that all the neurons in my simulation do the same as the neurons in the actual worm
mm.
in any situation, like
Yeah.
across a large array of behaviors. But then there's a second piece, which is I want that it produces the same behaviors in all external situation. It should behave in the same way if it's hungry. It should behave in the same
Mm.
way if there's sweet smell. It should
But
be
wouldn't that be sort of automatically fulfilled if the first thing was fulfilled?
no. It could be that all the neurons on the inside are strongly correlated with
Mm.
in the simulation with reality, but that it doesn't hook up correctly to the external world.
Okay. So it's like the input and output from the whole
Exactly. You
circuit.
need to get the inputs and the
Mm.
outputs right. And
Okay.
then the last thing, which is probably the most difficult, is I want that any perturbation should have the same effect
Mm,
as in the real C. elegans. Let's
mm,
talk about what we're talking about. Like take a neuron out. The
mm,
simulated worm without that neuron should do the same thing as like the real worm without that neuron.
mm.
Stimulate a few of them. Take a drug that like stimulates or depresses a thought of the neurons. Kind of like what I want is kind of this full kind of perturbative truth in the system.
Wow. Yeah. That's that sounds sounds exciting and quite challenging also. But but one thing which is like key to your, I mean, you have thought a lot about causality in general, right? I mean, I think you wrote a paper with my good colleague Mikkel in Oslo, right, about sort of causality, some ideas from economy, right? Sort of how to,
Indeed,
yeah.
yes.
So causality is like a thread through your, well, at some of your research, at least last year. So that's a key component, that you need to be able to causally stimulate or to stimulate neurons inside this sort of like C-elegans, so that you can sort of get sort of the effects of stimulation of one neuron on all the others or something in a systematic way. Is that correct?
Yeah, it's almost like if you want to know how a system causally works, you have almost no choice. And Julia Paul gives you the exact criteria, but to actually stimulate the systems.
Mm-hmm.
The reason for that is exactly that confounding that I mentioned earlier. Like the neurons all interact with one another. If I don't stimulate, they will basically all be moving in tandem with one another because they all influence one another.
Yeah.
Only by stimulation can I kind of discover this space and discover how they causally influence one another.
Mm. Yeah. So how then do you go about stimulating these 302 neurons?
Yeah, and this is another really cool thing about C. elegans, that we have established mechanisms to stimulate neurons. And it's just
Mm,
optically. You can say, I focus on a neuron
mm.
and it carries a molecular mechanism that converts light into
Some kind of.
stimulation currents. And by doing so, and Andrew Leifer has been pioneering that, you can basically go and focus on one neuron after another and see what happens to all the other neurons. So you can,
So that's like this channel rhodopsin things.
that's right. There's
Yeah.
multiple ways of doing that, but it's
Mm,
exactly that idea.
mm.
And so what's possible now is basically record from all neurons at the same time. There's
Mm.
this technical difficulties. No? Let's say, for example, the visual field of most microscopes is too small.
Mm.
There's technical problems, but in general, you can record from all neurons at the same time, and then you can stimulate random access, where you can say, I want to stimulate this neuron and now this neuron, or like this set of three neurons,
Mm.
and we have these methods now.
So you need some. Then you need some kind of two photon stimulation to get high enough precision or spatial precision, or
Yeah, so
yeah.
so so I've been talking with a lot of people, and there's two very different approaches you could think of. Either you use two photon holographic stimulation of neurons.
Mm.
The alternative is you can use molecular paths, where you can say, let's take every neuron and let's have the neurons basically choose a random subset of the 300 neurons, so that only that random subset carries the opsin, and then you can therefore, like, without any focus, stimulate those neurons, but you only get like one neuron per animal.
Mm. Oh,
So that's why there's an interesting space there.
yeah. Because but that's sort of like you sort of, in order to do this in practice, you need to I mean to have like a full like a many animals, and like you need some kind of industry style, almost sort of mass production of data, and the idea is that like the animals are so similar that they can somehow, the data can be pooled.
Yeah, so that is the strong hope here.
Mm.
There's alternatives to that, but to the level that we can model the individuals at basically the synapses are drawn randomly from some subset.
Mm.
That is the most that would be easiest. I want to give you a flavor of why it's still so difficult.
Yeah.
No, so imagine you could like figure out the strength of one synapse by stimulating one neuron optically and recording from the other one. Now, like, how long would that take you? Maybe a minute, maybe a few minutes. It kind of depends on the parameters and how strong or weak that thing is, but somewhere in that range. Now, if I give you 10 synapses, how much longer is that going to take?
Mm.
Well, if they can be arbitrarily nonlinear, then
What do
you
you mean
need
10 synapses? 10 synapses from the same neuron, or
no, maybe 10 presynaptic neurons, one postsynaptic
okay,
neuron.
exactly. Mm.
No, then I can do the 10 synapses one after the other, in
Mm.
which case it takes me 10 times as long, let's say half
Mm.
an hour.
Mm.
But then the question is, shouldn't we allow them to do something nonlinear with one another?
Mm.
No, like if you
Yeah.
have 10 synapses locally in some dendrite, they
Yeah.
often do nonlinear things with one
Yeah.
another. Or let's
Mm.
say one of those 10 could be a neuromodulator that really is meant to change what the
Yeah.
others do. So in that sense, we need more than 10 times as much. We probably don't need that exponential space. No? Like in principle, it could be that you need 10 to the 10, and
Yeah.
then like you're
Mm.
basically hopeless. So,
Yeah.
but that gives you like the flavor of what we can and cannot
Mm.
kind of take care of. For us to be successful at figuring out what the neurons in C.elegans do, we need to assume that the neurons are not too complicated. And
Mm.
so what we'll basically be assuming generally is maybe we need like order 1,000 parameters, no, where some of them are the synapses and some of them regulate the nonlinear
So
interactions
1,000
between them.
parameters for the whole network,
No, for the whole neuron.
for the whole neuron. Oh, okay. So then it's like for so 1,000 parameters describing the, like how a single neuron gets inputs and from others, from presynaptic neurons, and also projects to others or something, or
Yeah, exactly. No, you can
yeah.
say why do we come up with roughly the number 1,000? So a neuron might have 30 incoming synapses, but a neuron has non-synaptic inputs from a lot of other neurons. No, like
Mm.
they have hundreds of neuropeptides that basically get expelled, and
Mm.
where neurons thereby have a they call it the wireless connectome. It
Mm. Yeah.
basically is how neurons have an influence on one another. So
Yeah.
you can say from that perspective, we need about 100 parameters because that's the number of things.
But you still think you can map out, in principle, this, what do you call it, like the, what do you call it? This the floating, no, what do you call that? The fancy name, the non-synaptic connectome,
The wireless connectome.
wireless connectome. Yeah,
Yeah,
but you can still get it from stimulating presynaptic neurons.
yes,
Is
yes.
that the hope?
Keep in mind that the wireless connectome, just like the wired
Yeah,
connectome, comes from synapse from neurons to other neurons.
that's true.
No, so you can say if I can stimulate the presynaptic neurons, I can also find out what their wireless influence is. How do we expect to be the difference? No, like
Yeah.
you expect it to be a relatively fast, you expect a very fast response for gap junctions, and you expect a somewhat slower
Mm.
influence on chemical synapses and a relatively slow time course of like
Yeah.
these wireless connections.
Mm. Excellent. Yeah. Cool. So because you even sort of, so that is sort of like, I think you introduced this term interactome. Yeah, yeah.
Yeah, exactly.
So that's sort of beyond the connectome, because connectome is, I guess, just whether there is a connection between two neurons
Yeah,
or
no,
not,
no, like
or
computationally, it's a function. You can say the output of a neuron
mm,
depends on kind of all the things that influence that neuron. That can be external stimuli, that can be potential drugs in the system or something, and that can be the other neurons. And what you can do is you can stimulate all the other neurons. So you're trying to basically, in terms of neurons, basically get at the how does a neuron's output depend on the 300 other neurons. That's kind
mm,
of the core of that. Now, of those 300 other neurons, we expect that there's a good number that have no influence at all.
mm.
Maybe those that have like no neuromodulators that are important and no synapses onto the neuron of interest.
Yeah.
And out of that space, you kind of get to see why we kind of come within up with the number of 1,000.
1,000. Okay, so now we have, let's say we have all this, you have the interactome, all these connections that you have sort of mapped out optically, stimulated optically, and measured optically. And then now you need to put this all into a model. Or I guess what you have sort of measured now is what you call the input output relationship, or
That's right. No, you at this point of time, you should know what the output of any neuron is as a function of any possible input it could receive.
mm, any kind of synaptic input, or any
Or
or
non-synaptic input,
non-synaptic input,
wireless input.
any kind of connection between two neurons by any mean whatsoever.
That's,
gap,
that's,
ju-
that's right. That's
gap
the hope.
junctions, or wireless, or synaptic. Okay, good. So you have that, right?
That's right. And at that point of time, you
Mm.
can put the input-output function of every neuron into a simulation,
Mm.
and you want to like recalibrate it a little bit to get rid of accumulating errors, and then you're gonna
So
hook
what
it
does
up
that
with.
mean? What do you mean by that? So
Okay,
accumulating.
so imagine you just focus on individual neurons. I give you a
Mm.
model of neuron 1 and a model for neuron 2, and so on and so forth.
Mm.
What you usually have is if you build models out of components, let's say I now want to simulate all 302 neurons based on my model of every individual neuron that I have. In
Mm.
those cases, there's going to be some level of errors that accumulate by virtue of like us making small measurement mistakes on individual ones. So what you will almost always do in those cases is you will refit the data on the overall
Mm.
data, where you could say now I not don't just have one neuron, but we have all 300. We start with them, maybe what we learn about individual neurons, and then
Mm.
you will allow a little bit of like refitting just to
I
get rid
see.
of these accumulating
Mm.
errors.
Yeah, and now sort of and then the question. Okay, so and then you haven't spoken, or you haven't told me what kind of model you're thinking about yet. I mean, you're not thinking about sort of like biophysical models or integrated fire models, or what are you thinking of?
So integrate and fire neurons probably don't have the capability to fit well.
Mm.
So I don't, in particular, given that C.elegans neurons by and large don't spike.
Spike.
So
Yeah, no, that's true.
I don't think that that's the way to
Okay.
go. And neither
Mm.
am I thinking about linear models.
Uh,
I'm thinking of two different approaches, and I'm currently unsure which ones will be best. No, and one possibility is I can basically set up a biophysical like model and
mm.
I can make that differentiable. So I can have a JAX implementation of a differentiable neuron.
Jax. What is that? That's
Oh,
the.
JAX is it's basically like Python NumPy, only that you get all the derivatives very easily.
Okay. Okay.
And
So it's like Jax is not the. I thought it was like the book of Jack and Noble and Zed, but this.
no,
So
no,
this.
no. Like that's
Uh,
just a technical thing.
okay. Very good. Some kind
Basically,
of differentiable model.
if
So
you
you
if
can
you
think of it maybe as some kind of firing rate like model, maybe.
well, I would rather say it's really like kind of a nonlinear biophysical model that you
Okay.
want to optimize over.
Okay.
And then you need something, you know, like if that model also has thousands of parameters, you need like an efficient way of fitting them. So
Mm.
traditionally, the way we fit biophysics is by basically making proposals and seeing how good they
Yeah.
make us. I don't
Mm.
think that scales very well in that
No,
sense.
that's
So
mm,
so in any case, that's one possibility. There's also
mm,
plenty of work that shows that such biophysical like models of neurons can basically fit anything. So that's kind of good and bad.
yeah.
Like it's good for fitting. It's maybe bad for interpretation. So that's one thing. Uh, but then the alternative is, of course, to go into deep learning space. You know, like you
Mm,
can say a neuron implements a function. um, we
mm,
will have large data sets, terabytes worth of data about like neural activities. We
mm.
might be best served by using today's systems that are good at dealing with terabyte sized data sets. And they're in the context of deep learning, just like transformers with clever coding schemes are probably the way to go.
So that, but that's typically based on this ReLU units, right? Isn't that sort of like some kind of linear, non-linear, what's called threshold linear
So I
neurons?
mean, like transformers, we built out of complex attention-bearing pieces,
Okay.
but
Okay.
yes, not
I'm
like
not. I don't know this field too much. Yeah. Exactly. So.
ReLU is traditionally a transfer function that's very popular in machine
Mm,
learning. It's very fast to implement.
yeah.
I think in various domains like Swish works well, and then there's of course all kinds of hybrid systems that have all
Swish. What's
kinds.
that? That's I. That's some
So
kind of.
so ReLU is only once differentiable. No, like
Yeah.
it's basically piecewise linear.
Yeah.
Um, there exists version that you can differentiate more times, and there's
Okay.
cases where that's a really good idea.
Okay. Okay.
And so there's a whole host of transfer functions that people
But
use
but
these
the
days.
thing is that they should be like some kind of should be some kind of learning rule that is sort of some backprop learning rule. Is that
That's right. Now, like
yeah
you want to get good fits to your neural
uh,
data, and you will want to allow for like a wide range of nonlinearity. So one
mm,
good way is of doing that is just use like a vanilla large deep learning system and
mm.
fit it to it.
Because then that's the thing because you need something which is where you have this backprop learning rule unless if not you're sort of back to this optimization that you have in these biophysical models, right? Isn't that
Yeah,
true?
like in high dimensional spaces,
Mm,
you simply want to use an optimizer that uses gradients. Why?
mm,
Because you'll converge much faster.
yeah.
And why does that matter? Despite 300 neurons sounding like relatively little,
Mhm.
it's still a very large number of parameters that you'll need to fit if you have parametrized models.
Yeah,
But
because you said like 1,000 parameters and like 300 neurons, then it's like then we can do it 300,000.
yeah, but that's the thing. So because we don't know the nature of those
No,
parameters for
no, that's
sure,
true. Mm,
we might be better
True.
solved by using a model not with 300,000 parameters. And like that model with
True.
300,000 parameters would almost suddenly be hard to fit. And
Yeah.
instead, you'd use a model with 3 billion parameters.
Okay. Yeah.
And then you can at least efficiently fit it.
Yeah. Because you need these parameters must be sort of you must end up they must some of the model must be formulated so that you can get this backprop learning rule, right?
Well, that's one thing. But like above all, if I want to fit in high dimensional spaces, I need to
Mm.
be in a domain where I'm not getting stuck in like horrible local minima.
Mm.
And the general finding is that by having heavily over parametrized models of the
Mm.
kind that we use in deep learning, you'll be able to fit to highly nonlinear
Yeah.
functions without getting stuck.
Yeah, exactly. So that is sort of like this idea. If it's very high dimensional, there's always one way down in this high dimensional space. Is
That's at
that?
least one way of thinking
Yeah,
about
I
it.
don't know.
Exactly.
Yeah, that's yeah, I don't know if it's correct, but that's this sort of yeah. You must be very unlucky to be have like a thousand dimensional a value in all thousand dimensional. Anyway, that's not important. Okay, so that's sort of the approach. And but one thing I've been thinking about this because I've been sort of interested in mammalian systems and networks there, and there I have the feeling that it's in some sense in the future when we know these sort of like and these plasticity rules and the learning rules, things that the number of parameters may go down because you don't need to fit actually conductances, channel conductances. You that is sort of trained by some yeah some like if learning rules which are so I mean is so
Yeah.
this here you think that synaptic plasticity is not so much part of this idea you have yet or
So I don't think C.elegans has like the level of plasticity that us humans show. Like
mm,
the C.elegans doesn't probably represent really complex world models. But I wanted to
mm,
push back a little on that. So
mm,
the question is, where in neuroscience should we expect simplicity? Now you
mm,
can say C. elegans is like small enough that we can probably reverse engineer it without appeal to simplicity. But
mm.
let me push a little bit back on
Well,
your
you have
notion
to
of
add some kind of simplicity, right? I mean, there's always I mean at the molecular I mean the molecular level, ion channel level, then you have a whole world below there, right? But
that's
simply that's
this
right.
right now. Like
So, but
we
but
we
relatively speaking,
we hope for simplicity at the neuronal input output function level.
yeah.
Now you can say people who study the mammalian system, where can they hope to find simplicity? And you can say a lot of
Mm,
people traditionally believe that they find that in the region to region mapping. You know, like primary visual cortex
mm,
gets you Bayesian gratings and
mm,
complex cells, and then V2 puts them together into curves, and V4
yeah.
recognizes the cat. No, and then
Mm,
IT recognizes the cat or something like that. That's like the
mm,
traditional model where like the simplicity was meant to lie at that level. Um,
mm,
but if you live in a world that's truly plastic, you can say whatever you see will basically be smeared out over millions of parameters. Now, my hair's blue. You've seen me
mm,
now. There's going to be lots
mm,
of synapses in your brain that kind of carry like a tiny bit of Conrad's hair being blue. So,
mm,
in that sense, that idea that computation could be simple seems unlikely. And what you already said
mm,
is like, well, maybe learning is simple, but like, and yes, that might be true, but it's
mm,
only simple once I also give you the statistics of the world. Now, the
mm,
statistics of the world for a mouse is like remarkably complicated. Not like they live these like rich lives, at least if we don't keep them in their usual cages. They live rich lives full of like really complicated stuff. And
mm,
we shouldn't expect in a world full of plasticity, if brains have plasticity, then the complexity in the environment of the mouse
mm,
starts drifting into all the synapses that it has, making the synapses impossible to interpret. So you can say the process with which a mouse is made by the world might be simple, but the resulting mouse is still very, very complicated.
mm. But could it be? I mean, There was this. I read this book by called Critical Problems in Physics. So John, this is from '97 or something. I don't know if you've seen it. It was John Hopfield wrote the like this wrote the chapter, and he made this point that if you actually use C.elegans, right, it has like you said, 10,000 connections, and the size of the what was it? The size of the DNA is like whatever about 10 to the 8 bits. So he said while so it's much more it's like 10 to the 8 bits for 10 to the 4 synapses, something like that. While in humans, like 10 to the 14 synapses, while the DNA is only like 2 orders of magnitude larger. So he's saying that like the C. elegans could be like a yeah, so I mean, in C.elegans, the DNA could sort of hard code in the connections, while in mammals it has to do it otherwise. So maybe I'm thinking maybe the basic while the basic unit for C.elegans maybe could be the neuron. Maybe for mammals it's more like populations of neurons, which are then self-tuned to actually be the effectively the neuron, or I don't know. I'm just
Yes,
thinking out loud how
yeah,
similar
yes,
yeah
yes. No, so let's push a little on that view. No, like
yeah
there exists a meme which is that basically the most complicated algorithm is C. elegans because basically evolution put all that complicated stuff into it,
mhm,
and then zebrafish with its 100,000 neurons is simpler because it starts to like learn all over the place. Mouse is simpler yet because it implements high level like
mhm,
learning circuits, and the human is the simplest in the world.
mhm.
Now let me push back a little bit on that on multiple fronts. So
Sure.
let's first push back on the evolutionary side. So there's the first thing is evolution over time. Basically, we come from simple beings like C. elegans. So
Mhm,
there's no doubt that we inherit a lot of like their complexity. For example, we have C. elegans has hundreds of neuromodulators, and everyone's like, or like hundreds of neurotransmitters, and everyone's like, look, C. elegans is so complicated. It has these like hundreds of neurotransmitters. And you're
mhm.
like, guess what? Mouse has them has more of them. You know, like
Yeah, yeah, yeah.
so that's the first thing. So like
Mhm,
over time we come from these beings, so there's no reason to
mhm,
believe it's simpler. The other one um, is from what you should be expecting. You know, like what is
mhm,
the best normative solution? Now you can say if we look at modern deep learning, it's not that these systems are getting easier; they're getting more complicated. No, it's not that these systems basically started with like something very complicated and now they just do gradient descent.
mhm,
No, we have these intricate hierarchies with a lot of structure, a lot of initialization, a lot of all kinds of aspects of that. So,
yeah,
if anything, and you can say we're now heavily leaning into self-supervised learning, which is basically we exploit aspects of the world to learn better,
mhm,
and there's no doubt no like we have short life still relative to what we need to learn. Like we live
mhm,
100 years. Like most important decisions in our lives don't happen 10 to the 6 times, they happen like once. And
mhm,
so therefore we are forced to operate well in this like heavily data constrained world, which is, you're right, no, like it only has 100 times more DNA, but
mhm,
that DNA will be chock full of stuff, which means that we shouldn't be expecting us to be able to like have a few simple principles that describe all of it. It will actually
mhm,
be like C. elegans, only worse, because it
mhm,
will also have like learning, and it will not just have learning like maybe gradient descent. It will have like
mhm,
circuits for learning and approximations for learning that are faster and that
mhm.
save you samples and all those
Yeah.
things.
Yeah, but I guess, I guess the one thing is what is simple. I think it's none of this is simple, whether it's, but at least to figure out. But I guess the question is sort of if you sort of, I think it would be fantastic to sort of to understand to be able to reverse engineer the C elegance. I think it would be a giant leap forward for the field. But then I guess the question is sort of how much could we expect the same principles or what principle do you expect to sort of to also apply that we'll learn from C. elegans also apply for mammals because it's like well it cannot be hard coded in the same way, maybe. And or so, what do you think of that? Or this is just speculation, of course. I mean, the first thing we have to understand C.elegans, and then
Yeah, there's no doubt. So fast, I mean, like maybe zeroth order.
mm,
The methods, some of them will generalize to bigger systems. So with C. elegans, we're very actively interested in finding shortcuts to reverse engineer
mm,
systems. And we will need those if we want to ever reverse engineer something like a mouse. So that's the
mm,
first. But then you can say the principles that are there, to which level what will carry over. And there can be like principles like localization, notion of modularity, maybe certain aspects of learning algorithms,
mm,
maybe kind of things about the flow of information through systems. Now, you
mm,
expect evolved systems to minimize the amount of energy per bit
mm,
computation or
yeah,
something.
mm,
So there's
mm,
arguably will be a lot of them that will be shared with us.
mm. But if you think, just thinking of you had looking at think about this CNN, like the deep networks, convolutional neural networks, for example. If you had two identical neural networks trained at like the same images, but like maybe with different initial conditions and maybe different order of the right, you'll end up with two equally functioning networks, presumably, but the synaptic weights would be different. And if you run this sort of like activation thing on that, that wouldn't really work, right? Because the neurons are not identical.
Yeah, and let's be clear: if you train two human beings
Yeah,
with
yeah,
the same initialization and all that,
yeah,
you will
yeah.
also
So
obtain
it's like,
two
yeah,
radically different nervous systems.
yeah, yeah. So that's, but I
No,
think, I mean, yeah,
but I think I interpret that in this direction, which means that in a way we carry information about our whole lives with us, no? Like,
mm,
which means that your brain must be different to mine, no? Like
absolutely.
we share parts of our worlds, but we also like experience very different ones.
Mm,
And that then highlights multiple difficulties, no? Like, it means that no description of your brain can be
yeah,
complete without
mm,
all the idiosyncrasies that made you you, no?
yeah,
Like,
no,
and the same thing about my brain.
mm,
And which means that it arguably can't be human understandable, no? Like,
mm.
because how could you summarize all the things that happened in your life?
Oh, but I guess that's also a question now into sort of what does it mean to? I mean, we have done some work with people at the Allen Institute where we looked at like data from 50, 60 mice, primary visual cortex, and of course they are different too. But that at some level they're different, but at some level they are similar. And I guess that's also the same if you look at
It's, it's
two
true for
neurons, which everything is different at some level if you just go deep
But
enough.
but let's be clear. It's true for neural networks. You know, like if you
Mm,
look at multiple neural networks, they all have GABA-like receptive fields in the early stages of their visual hierarchies.
mm,
They do have the salient grammatical concepts represented in the same way as humans represent
yeah.
salient grammatical concepts. So
Mm,
so at some level, we can play those
mm,
games. So, like,
yeah.
what's the represented information?
Mm. Okay, so let's see that you are successful. Now you have this not even like beyond digital twin. It's like the it's like this perfect replica in silicon.
Yeah.
Then 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 what do you do? Some people would say that that's just as hard to understand as the real roundworm.
Yeah, so let's maybe approach the word understanding, because
Mm,
neuroscientists love throwing it around, but they tend to be very imprecise of what they mean.
mm,
So you can say there's one definition of understand, which is what we will deliver, which
mm.
is a simulation that's correct and actually works. That's one definition of understand. But
Yeah.
it's not probably what, like, I think, and you and me in our heart of hearts mean with understand. Now, like, it
Mm,
doesn't it doesn't at all allow you to say understand, like describe it to me. Like I let's say the simulation runs on my
Mm,
computer, happy
mm,
C.elegans swimming in a digital gym. I'm
yeah,
gonna call you and I'm got it. Like here's
yeah,
how it works.
yeah,
That's not gonna happen in that sense.
no,
Why? Because it will have lots of parameters, hundreds of thousands or
mm,
billions of
mm,
parameters.
yeah,
And so you can say there's a notion of explanation or understanding that we're not delivering.
mm,
And there's multiple answers that I have to that. So the first thing is that level of understanding might not exist.
exactly.
So let me highlight what I mean with that. So evolution gave C. elegans all kinds of crazy abilities, including getting away if there's no more food. It's not that C. elegans is like, oh yeah, my plan number 37 is if this is happening, then I gonna like activate this thing.
Uh,
It just is.
mm,
And because we can't enumerate the many, many ways how bad shit can happen to like C.
mm,
elegans,
mm,
um, we can't fully ever grok that or describe it to another human being. No, like we
mm,
couldn't describe the behavior. It's like I could be like, okay, and if there's nice yummy food, but someone's about to parasite you, you better
mm,
get away from the food.
mm,
And you kind of it grows like exponentially about like how different combination of configurations make you do different things,
mm,
and evolution built all that into it. So
yeah,
at
mm,
some level, and the same thing, no, like we can't like simulate like another human being fully and
absolutely.
understand them because they're different. Like you have a different life to the
Yeah,
life I have.
that's
And
true.
so in that sense, so like first you can say maybe that level of understanding doesn't exist. Now you can say if that level exists or not, there might be aspects of it that we can understand. And now
Mm,
you know what I'm gonna do is I'm gonna just basically say, look, if we have this working simulation, we at least can mine it for principles that aren't
mm,
nonsensical. We can kind of say, okay, A, everyone, what are the principles you think there might be? Okay, let's test it. Like maybe
mm,
maybe they're right. You can say we can do algorithmic hunting for that, where you can say, what is like a loady description of C. elegans that carries most of the variance? And I can say like, actually, you know what? Like, there's these neurons, and not these neurons, and they're often like all active at the same time, and so much so that in everyday
mm.
life it explains 90% of the
Yeah, yeah.
variance.
Mm,
No, like, or we could say, and here's a little task, and during that task, it's really just these three neurons that do
yeah,
it. No,
yeah,
so there's like these localized principles
yeah.
that we
I guess.
all want to get at.
Good. I guess we are a little bit actually when it comes to one of the things I'm most impressed about is like numerical weather forecasting, where you can sort of compute the weather and more accurately. And I get, but I don't really know how much you can sort of what much. I mean, even meteorology can say about why did it rain yesterday at 2 o'clock. Well, and they can have like some principle why there was a cloud coming with low pressure there. So I
Yeah,
guess so. There, I guess you have already this advanced model, and it's a question: do these
yeah.
concepts like this gentleman or like these concepts we can understand, are they really useful? And maybe sometimes they are.
And I think it's kind of like deeply
Uh,
frustrating, like look, like a defining moment for me was at INI where I was a PhD student.
I and I.
Kevin,
Where's that? That was in.
Institute of Neuroinformatics in Zurich.
Mm,
I remember being in the lab with Kevin Martin, who was doing these experiments, like electrode in the brain, in a visual
uh,
cell, and he had them on on the whiteboard. You then project with the light, and you like move, and the cell goes like,
uh,
and there was like this
uh,
vision of like, wow, like that stuff is understandable. Maybe this is the way we like can understand us humans, how we like operate, how we think, and all that.
uh,
And me like struggling for like literally decades on that
mm,
issue, I think I'm approaching that place where I'm reevaluating what the things are that we should describe it in terms of. And for example, for me, if I can give you like the learning rule for that, where I'm like, okay, look,
Mm,
first order approximation is trying to like optimize roughly this notion of world understanding. And then like, yeah, like all the synapses in your brain, Conrad, are like moving to the right direction in that sense.
mm,
And that describes like a lot of the variance of like the learning process. That's something that like I think I would be very happy with. But there was this time that I deeply feel in me still, where I felt like I would like to understand how I think in terms of like how the neurons interact with one another.
mm.
I
Yeah.
just don't think it's possible.
No. 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 Oh, it's difficult to say how close yeah, whether we get this. Yeah. I guess sort of if you yeah, I guess it's unclear, right? Even if we get this like in weather, I mean, or even when people talk about like the stock market, right? It's a little funny to talk about the people talk about when the stock market goes up. Well, they like the commentators on TV say, well, I mean, people were sort of investing more than selling, essentially, right? They're just like saying the same thing, right? So this like whether there are concepts, and sometimes there obviously are concepts like this when it comes to weather, that there's like low pressures outside Bergen in the western part of Norway. That's going to mean rain often in some kind of. So of course there are these. So you have to explore this concept, right?
Yeah, no, look, there's no doubt that there's meaningful partial summaries and more meaningful partial summaries still to be discovered. No, like, there's no doubt that in primary visual cortex, there's more visual activity than auditory activity. No,
Mm,
like, but
yeah.
by now we know that there's actually like some auditory and a lot of behavioral
Yeah.
and a lot of social activity and so on and so forth.
Mm,
But there are these things that are like true and described data. I'm not
yeah.
denying that. Not like in a
Mm,
lot of neurons in primary visual cortex are
mm,
strongly tuned to orientation. Like
yeah.
these things are true, and yet they don't describe how the system works in
No.
its totality.
Yeah. So
And that is like a weird intellectual struggle how to like think about those
yeah.
systems. And I think it's in everything. Like it's in weather prediction. Like we're getting better and better weather prediction models. We don't understand them because we're
Mm,
like building deep learning systems into at these
yeah.
as we
That's
speak.
also true. Yeah. So they're
Eh,
doing yeah, all kinds of tricks.
and
Yeah.
and we are decent at predicting company performance, and we are decent at predicting future successes of professors. Now, like we've done some of that in the past,
Mm,
but it doesn't describe how it works. It's just good at predicting. So at some level, even those things that are good at predicting
yeah.
don't tell us the truth of what that means.
No. No. Yeah. So I guess with a new develop well, I know new the rapid use of AI, this could going to be more and more of this, right? That's sort of like this kind of limited on the predictive predict yeah ability to predict but not explain.
Yeah, and like if you look at machine learning, it's really not doing well on causality. So
Mm,
it's really good at predictions. It's really not good at understanding why
mm,
the things happen.
mm. I want okay. So another point. I mean, this kind of this project, this reverse engineering. I guess you could sort of you could build a model to have this as like a model of these 302 neurons with some kind of dynamics, and see to what extent you could sort of simulate the whole reverse engineering project, right? And test this whole approach in a virtual environment first. Is that something
Yeah, and
is good?
of course we've done that. No, like we
Yeah.
started with like a simple simulation of interacting neurons,
Mhm.
trying to see how much of what kind of data we'd need to
Yeah.
reverse engineer it.
Yeah.
And that is kind of these things are how we come up with our numbers.
Exactly. Excellent. Yeah.
And
That's like
so our hunch is we'll need order 10,000 experiments.
exactly based on yeah, exactly. Because I think this is something which has been I mean, I mean, this thing of making model-based benchmarking data, sort of for to test all kinds of data analysis method. That's something where we need to do better as a field, I think, because this is, of course, even if it worked for some model-based simulation data, then it doesn't mean that it works for the real thing. It could be other things, right? But at least if it
Yeah.
doesn't work for the model-based data, then you have to think twice maybe about the whole approach.
So from my perspective, that's one of the beautiful things about the C.elegans project,
Mm,
which is there's a large branch of neurostatistics that aims to figure out causality based on correlational data.
mm,
I'm skeptical about that for purely statistical reasons,
mm,
but success at the C.elegans project would allow those folks to prove that the algorithms actually work, or it would allow me to prove
mm.
that the algorithms don't.
Exactly. Yeah, exactly.
But
Yeah.
it promises to kind of put that branch of neuroscience onto a cleaner
Mm,
logical
yeah,
basis.
yeah. I think that's something we really can do, and we should do as a field. I think sort of to do this kind of tests on of approaches on benchmarking data where we know the ground truth. So
And that brings us back to the original microprocessor paper. No? Like
yeah.
the reason why we did that is to
Mm,
have a domain where we have ground truth. And of course, a lot of people said, "Oh, it doesn't meaningfully work on the microprocessor. It would
mm,
have worked wonderfully if only we had like a real brain." Well,
okay.
the
Yeah.
elegance brain is maybe not the brain these folks are
No,
thinking
but I
of.
mean, we have models for I mean, cortex models and sort of like I mean, like well, for example, from the blue brain model, which is sort of like maybe not, even if it's not very close to biology, I don't know, but nevertheless, it's sort of a model where you can produce benchmarking data. Same thing with the Allen model for the mouse visual cortex.
That's right. And do you know why no one is using those models as benchmark data?
I don't really know. Well, we sort of used the Allen model, but yeah, I don't really know. Was it a question, or do you have the answer?
I have a theory.
Okay, come with the theory.
So my theory is that people don't use those real models much because it doesn't work.
Yeah, yeah, that's very true. Yeah, but maybe
No,
it's also
but
yeah.
like that's the like it turns out that inverse prompts, and like we're generally talking about inverse prompts. We have
Mm,
observed data of some
mm,
sort. We
yeah,
have an underlying system of which we know some things
yeah,
that makes the data, and we
mm,
want to solve the inverse prompt. And
mm,
inverse prompts are almost always hard because there's dimensions that are easy to estimate, and
yeah,
there's other dimensions that are hard to estimate. And even in like just linear fitting, like
mm,
there are usually you have some dimension input dimensions that have high variance and others that have very low variance. So if you
mm,
do PCA or something and you plot the power of the kth component, you like usually
mm,
find like some kind of a power law. And that means that there's some dimensions that are very easy to know things about, and a
mm,
lot of dimensions that are essentially impossible to know
mm.
anything about.
Mm. But I guess also another thing is that, like, I mean, when you're fitting sort of, I mean, the traditional, when you fit low dimensional models, meaning small models with few parameters, then you typically have a unique solution. While this biological high dimensional model, many parameters, it's not a unique solution, and that's maybe probably more like a feature than a bug. That is actually some self organization of the biological system to have. I mean, there's like many ways to implement the function. So it's not about finding a unique. At least I heard sometimes this critique against this sort of well, large scale network models, that is not a unique solution, and I think that's sort of a there are
Yeah.
many challenges with that, but that's not the goal either.
For,
We have
for,
to learn to think about these things differently, I think.
for every neural data set that anyone in neuroscience has ever found out about, apart from like some very, very, very simple systems,
Mm,
there is an infinite set of biological, meaningful,
yeah,
potentially realistic models that will predict exactly that data set
mm,
within the noise that's there.
yeah,
Which
mm,
means that the neuroscience that we do doesn't actually inform the mechanisms that we
mm,
want to talk about, which
mm,
puts large branches of neuroscience onto an epistemologically really difficult spot.
mm. So, I mean, now what is required to do this project like reverse engineering? I mean, there's always been this open worm project, which has been on C. elegans. Can you tell
Yeah,
about that a little bit? Sort
so
of,
so so we're I put together a project for a grant recently,
Mm,
and we're collaborating with the Open Brain project, so the Open Worm project. So the Open Worm folks want to simulate worms, and I think they weren't so aware when they got started that basically the necessary experimental data just isn't there yet.
mm, yeah,
It they built a wonderful simulator for the body of the worm called Cybernetic,
mm,
but the data about what's going on inside of the worm in the nervous system is just very, very partial. And the way the scientific literature is written makes it near impossible to basically put what we learned from there into like one cogent model
I
because it's
see.
all just so very, very partial. So I think like that lack of completeness is the big problem. Now, like
So the
if you
data missing was like synaptic weights and the properties of the neurons and stuff like that.
oh yes, we didn't know at all what the weights are between neurons.
Mm,
Like the typical study in most branches of neuroscience is of that nature. We look at one small behavior, and in that one small behavior, we find a set of neurons or a brain area that is strongly affected by that. That does not reveal how they do that. And that's kind of like outside of what no, like it's usually hidden behind like a strong specialization idea where it's like, okay, I find three neurons that are strongly correlated with that. Therefore, these neurons must be the ones doing it.
mm, mm,
And then it's usually not spelled out how they do it, and it's usually not spelled out what like the whole how the
mm.
rest of the system interfaces with it.
Okay, so what's your plan now for? I mean, you this is sort of like a well, we're sort of approaching the end of the podcast, so I guess I mean you for to get this reverse engineering project off the ground, you need what you need. I mean, collaborations, people to join in. Should they sort of write you an email, say I think this is really cool. How can I contribute, or how can I fund you, or maybe
Uh,
what is needed?
yeah, so I wrote together with the dream team in that area a wonderful proposal to
Mm,
the Simons Foundation. We'll
mm,
have to see how they think about it, but
mm,
it feels to me like it's a transformative approach because in almost everywhere in neuroscience, you need to make these strong assumptions about the part of the system that you don't see. Those strong assumptions are generally not justified. Um, I feel this is the first system where we can really be sure about how we do neuroscience because we'll have completeness. No, we'll have observational completeness, see all neurons. We'll
mm,
have perturbation completeness, perturb all neurons, and we'll
mm,
also have this full set, rich set of behaviors that C.elegans has.
mm,
So that team consists of we need to build microscopes basically, and we need to build computer vision pipelines, and we need to use molecules to better figure out
mm,
how neurons might be interacting with one another, and we need to build models for that. And so
mm.
I think this will be a large collaboration across lots and lots of labs. And
So,
so yes, if people have ideas about it, I'd love to hear about it.
but I mean, in terms of let's say microscopes and the stimulation thing, when you do, so how many setups do you need, and how long do you need to run? Is it something you do at UPenn, or is it where is? I mean, I have no. I mean,
Oh,
if you look
oh,
at the Allen Institute, what they done for the mouse visual cortex, that's really like an industry style, or like a normal, like a large
yeah.
number repetition of the same system. So it's like is it that kind of operation you're thinking of, or?
Yeah, I'm thinking we should have like two, three setups that run 24/7 and
Mm,
ideally run multiple worms in parallel.
exactly. Yeah.
But altogether, yeah, it's not a cheap endeavor, but it's just the obvious, from my
Mm,
perspective, the obvious next development in neuroscience.
mm. Yeah. Exactly. But so you say that the bottleneck is really the experimental recordings, or like stimulation, and doing that sort of at the.
Yeah, so I think at least the primary current bottleneck
Mm,
is scaling up data generation. And then on the theory side, building systems that are good at fitting will become important, but that only becomes important once we have petabytes worth of data.
I see. Yeah, I see. Yeah. Cool. So excellent. This was really exciting. I think this is a very exciting project, and we need some bold new ideas to to move to neuroscience forward. So thanks a lot for coming to the podcast, Konrad.
Yeah, thanks so much. This was a fun discussion.
Yeah, absolutely.